Hi, Nice Genes listeners. Sarah Lando here from Genome BC. Before we get into our special episode today, I have some exciting news I wanted to share with you, especially if you're in the Vancouver, BC area and love genomics and space. On October 6th, Genome BC is hosting the annual Don Ricks Distinguished Keynote Address at the Vancouver Convention Centre, and this year's keynote speaker is Dr. Christopher Mason. Dr. Mason is a NASA scientist and globally recognized genomics expert doing cutting-edge research on DNA sequencing in space, astronaut health, and how long-term space travel affects the human body at the genetic level. It's a rare opportunity to hear from someone at the forefront of space and genomic science. In-person registration is free, but space is limited. So head to genomebc.ca to grab your ticket before they're gone. All right, let's launch into our story for today.
This is your captain, Kaylee Byers, reporting from the Nice Genes mothership. So, you might be thinking, hang on, why are we in a spaceship? Or at least a very convincing simulation of one, obviously. Well, consider this an amuse-bouche of what's yet to come. We're just popping into your feed with a special Gene Short, a bite-sized science story to tide you over until our next episode lands. I'm sorry, hold on. I'm receiving a signal. It looks like it's coming from outside the ship.
Captain Byers, this is Sarah Lando from Genome BC, requesting permission to dock with a fresh new science story.
Impeccable timing, Sarah. Initiating transport sequence now. Hang tight.
Airlock secure.
Made it in one piece. Hello, Kaylee.
Always happy to have you aboard. I have a feeling you've got something stellar up your sleeve, or should I say a spacesuit.
You know it. As fun as this virtual spaceship is, I've got a story that takes us closer to actual stars. So Kaylee, space travel is starting to feel a lot less sci-fi these days and more like real life. More and more people, billionaires, are lining up to explore the edges of our atmosphere. And sure, these trips get all the attention. And when we think of space, it's usually the big flashy stuff. You know, exploding stars, supermassive black holes, galaxies colliding. But today's story, it's not about any of that. It's about something tiny, microscopic even.
Oh my gosh, I'm so excited to hear a story about actual space and not the famous people going to space.
Right? So our story begins with an exciting new discovery. Scientists recently found something very curious thriving in China's Tiangong space station. After doing some routine microbial monitoring aboard the space station, you know, standard stuff, they found an unfamiliar strain of bacteria on board. After sequencing its DNA, they discovered it was a brand new bacterial species never seen before on Earth. And the name, Naellia tiangongensis.
Okay, so they find this new bacteria, you know, just standard space cleaning stuff, and they slap a name on it, but what do we know about it other than its name?
Okay, so it's not totally alien. It's actually closely related to Earth soil microbes. Therefore, it likely hitched a ride to a space station, whether it was like on equipment or on a person. Perhaps the person was an avid gardener. Then it did what we all try to do best, survive, but in space. And since the space station isn't exactly its normal environment, this bacteria made some key adaptations, which have been revealed by genomic sequencing. However, that being said, there's still a possibility this could be a bacterium we just haven't identified on Earth yet.
So, okay, going with the theory that it was a bacteria from Earth that hitched a ride and then evolved some adaptations that were really space-worthy, what sort of evolutionary tricks did this hitchhiker have going on?
So the space station environment is no five-star hotel. It's extreme, with things like microgravity, increased cosmic radiation, and all that jazz. And that stress possibly caused the microbe to activate parts of its genetic toolkit.
Okay, so like, what's in that?
So, for example, stronger oxidative stress response. So its ability to handle radiation and oxygen damage, and biofilm-forming behavior, which is like a protective slimy coating that can help repair DNA damage.
Cool.
And this is really interesting because if this bacterium evolved these traits as a survival mechanism, it's likely that this process unfolded over months or even years not overnight.
Okay, so this is pretty cool. But I don't want to start stereotyping bacteria because, most bacteria are health promoting. They keep our planet functioning, healthy, and happy. And so this podcast is a pro-microbe podcast. But just thinking about the astronauts living on these space stations, they're in close proximity to this bacteria that we don't know all that much about. Based off what we know about other bacteria in this same genus, does it have any potential impacts on human health at all?
Good question. So as of right now, the bacteria isn't known to be harmful, but there are related strains on Earth that can cause infections in immunocompromised people. It could pose risk to astronauts or spacecraft materials if uncontrolled, though pathogenicity is not confirmed yet. I think what's really important is that we stay vigilant. We need to be mindful of proper sanitation in general and on space stations. This also really teaches us that we need to keep track of how microbes evolve in space, especially because these microbes might even be useful with the right engineering.
Useful? How?
So this kind of thing has actually happened before. For example, back in 2021, NASA scientists discovered 3 never-before-seen types of bacteria living on the International Space Station that they called Methylobacterium ajmalii.
Methylobacterium ajmalii.
Yeah, not an easy term to say, but these microbes are related to bacteria that actually help plants grow. So scientists think that maybe one day it could be useful for growing food on Mars.
Whoa, imagine if Matt Damon had those to help grow all his potatoes.
Yeah, exactly.
Potatoes for all.
Much better for Matt Damon. So these microbes, they're constantly adapting even in environments built to keep them out, which is pretty fascinating. And genomics helps us spot these changes, even when we can't see them with the naked eye.
I love that. Who would have guessed that on this podcast, genomics would have been here to save the day?
And that's why you're a DNA detective, Kaylee. So here's the thing. We are not going to space alone. We bring trillions of microbes with us, not just on us, but in us. Remember, we are made of more microbial cells than human cells. And some of these microbial cells may change in unexpected ways under space conditions. What we need to do is keep an eye on them and think, what are we bringing with us and what are we bringing back as well? Because the more back and forth of humans to space stations, the more opportunity for bacterial colonization. And it's all part of the story of space exploration, but we need to monitor it.
Wow. It's really not the journey to space, but the bacterial friends we made along the way.
Couldn't have said that better myself, Kaylee.
Sarah, what a treat, what a treasure to see you as always, and to have you on the pod. I gotta say, a star is born.
Oh my God, thanks for making the space to fit me in. How do I go back to Earth right now? Where's the door?
Oh my God, don't touch that! Ah! Ooh, yikes.
Airlock secure.
Well, that's it for now, space travelers. Thanks for tuning in to this Gene Short. We'll be beaming into your feed with a full episode faster than a shooting star. Until then, this is Captain Byers, over and out.